Okay, good morning students. So we are going to continue the chapter strategies for enhancement for food production, right? So the topic that we are going to discuss today is we are going to talk about plant breeding for disease resistance, right? We have talked about what is plant breeding, what are the steps of plant breeding, why we should go for plant breeding, etc. in our previous classes, right? So what we will be talking about is plant breeding for disease resistance. What we know is there are so many different types of pathogens like viral pathogens, it could be bacterial pathogens, it could be certain types of nematodes, right? It could be fungal pathogens. These pathogens are the main cause of causing disease in case of a plant. And whenever plants are affected by certain types of diseases, what is the result? The result is always crop loss, right? So diseaser karone ki hoy bahut bilak crop loss hoy no, and in order to prevent that crop loss, what we need to do is we have to produce certain plants that are disease resistant. I think we understand the terms resistant and susceptible, right? What is resistant? The eta pathogen adult plant jodi attack korise and the plant does not show the disease or the plant does not get affected by that particular pathogen that has invaded it, then we use the word what? That plant is a resistant plant. And if suppose a pathogen attacks a plant and the disease is seen in the plant or the plant gets affected, that is called as susceptible. So what we are talking about is, we are talking about that since there are 1,000 numbers of pathogens that are present and they are responsible for creating various types of diseases, plants bilakor ki hoy different disease hoy, ar jetia disease bilak hoy tetia hole ki hoy crop loss hoy. So what we want some disease resistant plants, right? Disease resistant plant produce korile, what would be the advantage? The advantage would be the production of food will increase, right? So plant allows it disease no hoy, then obviously the production will increase. So in short, what we can say is the food production will increase. Second advantage would be the application of or the use of fungicides, pesticides, etc. certain types of chemicals would decrease. Ami fungicides, pesticides by enuka dhoroner chemicals bilak ketia use koru jeta plant alor disease hoy, right? Jedi plant alor disease na thake tetia there is no point that we will be applying all these chemicals. So if we can make some disease resistant plant, then obviously food production will increase and the use of chemicals would decrease. Now whenever we are talking about disease, pathogen and the plant, right, there should be some factors because of which the plant is affected by the disease, right? Plant dalor disease kiho karone hoise kisman factors thakibo, and what are the three factors? We will be discussing about three factors. Basic details of discuss koribo na lage, but the point that we need to know is whenever we are talking about a plant getting affected by a pathogen, there are three conditions on the basis of which the disease occurs. What are the three conditions? Or the disease depends upon, it depends upon the host genome. Three factors upon depend kore. First ki host genome, second ki pathogen genome, and thirdly it is also on the environment that the disease will occur or it will not occur, it depends upon. So tinta factor upon depend kore. First is host genome. What is host genome? We are talking about the genetic constituents of that plant, right? Jundal plant or disease hobo, hey plant dalot disease hua karne kun responsible hobo? Genes could be responsible for causing that disease. So who is responsible for the disease? The genome of that plant or the host plant is responsible. Secondly, at a particular pathogen, at a particular plant affect kore. Again, we have to talk about the genetic makeup of that pathogen. So we are using the second factor, or we are talking about the second factor, that is called as a pathogen genome. And thirdly, plant genome you are talking about suppose we are talking about the plant jarniketa gene is responsible for causing that disease, and we are talking about the pathogen who has invaded our pathogen or jinto gene ase, that gene is also capable of causing a disease. But in spite of that, jedi environment factors bilak favorable no hoy tetia hole disease to cause no hoy. So ultimately environment also plays a major role in causing a disease. So from these three sentences, what we need to remember is pathogens or karone ba pathogen attack or karone ki hoy disease hoy, disease or karone crop loss hoy, crop loss no hobole we have to prepare disease resistant plant. Disease kyo hoy, but what are the factors on the basis of which we can decide? There are three factors, and what are the three factors? Host genome, pathogen genome, and the environment, and these three are always interrelated, right? Okol etar kotha porile na hobo, host genome is related with the pathogen genome is related to the environment is also related with these two factors, right? So ultimately a gutei tinta factors jodi favorable hoy tetia hole that would result in the causing of disease to the plant, right? Now, since we are talking about diseases, what we need to read from this section is there are certain plants or there are certain economically important plants in which certain types of diseases has been seen, or because of these diseases it has led to the crop loss, right? Kiman plants ase, bahut plants or bahut type of diseases ase, kintu NCERT jungita dia ase 5 numbers or 5 to 6 numbers, these are the things that we need to study. For example, we have some important disease of economically important plants. So we have a disease called as late blight of potato, right? Second disease could be loose smut of wheat, black rust of wheat, etc. So ebilak hol certain types of diseases that are often seen in these economically important plants, right? Aru etu nohoy je these are the only diseases, but from NCERT point of view we have to study only these, right? Now, what is the question that is asked from this part? Yet a question ahe, the questions are of various types. For example, if you are talking about the first disease, that is late blight of potato, question ahe, what is the causative organism of late blight of potato? In that case, you need to remember it is Phytophthora infestans. That is the first point, right? So late blight of potato, causative organism ki, or causative agent ki, it is Phytophthora infestans. Second question about this disease or any sort of disease that is asked is, hey, disease to juntu kotha koisa, whether that is a bacterial disease or it is a fungal disease or it is a viral disease, ne kunba nematode je cause korise, right? So whenever you read about the disease, duta bostu poribo laibo. First is who is responsible to cause that disease, right? Causative organism. Second is what is the type of disease, viral, bacterial, fungal, ne ki. Talking about this first one, late blight of potato, Phytophthora infestans. Obviously, what type of a disease is this? Bacterial, fungal, viral, kenuka dhoroner disease. We are talking about a fungal disease, right? So agent to monot rakhim. Another point about this is, it is also said to be the famine of Ireland, right? Kiswan sentence ami pori ahisu jeneke, for example, if we talk about terror of Bengal, what we remember about a specific plant that is called as water hyacinth or ecornia, right? Thik teneke ami ki janibo laibo etia, famine of Ireland. So famine of Ireland boli kole, which disease we are referring to? We are referring to late blight of potato. Now, as far as the causative organism is concerned, we talk about what are the symptoms. Symptoms or pora iman questions ahanai. Enuka dhoroner question ahanai je what is the symptom that you see in case of a potato plant when it is affected by late blight, right? So iman details nalage, just to have an idea. Late blight hole potato plant alot ki hoy, you have the leaf of the potatoes, or leaf pilak or lower side ot ki hoy, water or what you say is hydrolytic area. Water jama khale jenuka hoi jay no, ba pani eta patot jodi pani jama khai thake, he pani khini jeka karone swollen hoi thakibo, okoman damage leaf hobo. These are the symptoms of this plant. Symptoms of a generally question aha nai. So what you need to know is just to have an idea, late blight of potato, what is the symptom? You will see some hydrolytic areas in the lower side of the leaves, right? Which leaves? Obviously potato leaves, right? Causative organism gom palo, kenuka dhoroner disease, it's a fungal disease. Jedi fungal disease hoy, ami a disease to control koribo lagile, what we have to use? Obviously, we have to use certain types of fungicides, right? Fungicides are details of, when we are going to talk about fungi in the chapter biological classification, then we are going to discuss the proper names of fungicides, their composition, etc., etc. As of now, what we will remember is, since this is a fungal disease, obviously to control it, we have to apply certain types of fungicide. If you remember, body X mixture boli kiba ta bostu asile, we'll talk about it later, right? So that was the first disease. Second is loose smut of wheat. Loose smut of wheat or causative organism ki Ustilego nuda triti ki. Ustilego is again an example of a fungi. So loose smut of wheat is again a fungal disease. Control kenege korim by application of fungicides, right? And what is the symptom that you will see? You will see that the inflorescence would have some powdery black spots or dry cola powder or nisnake kisuman bostu dekha paba. Kod dekha paba in the inflorescence of the wheat. Now, what we understand by inflorescence? Inflorescence mane ki? Disease tour symptom to ami first code dekha pao, inflorescence or dekha pao, right? Wheat or junto inflorescence ase, tar ami ki dekha pao, jetia itu disease hoise, tetia hale he inflorescence to ami ki dekha pam je kisuman black color ba kisuman powder dry type substance inflorescence to ase, or as a result the inflorescence becomes dead, right? Now the question is, what is inflorescence? The collection of flowers is called as inflorescence, right? Wheat or junto portion ami khao. Next we'll come to black rust of wheat. Black rust of wheat. I mean, jetia black rust ko, no? What is the point that we remember? We always remember about Puccinia, no? Question hua hai, black rust boli kole, which fungi are we talking about? We are talking about Puccinia, right? Puccinia graminis. Jedi Puccinia graminis treaty ki monoto na thake, Puccinia graminis is a word that you remember, and the easiest way to remember is black rust hole khodai Puccinia hi hobo. Talking about Puccinia from the fungi part, another point that you need to remember is that Puccinia is always a heterogeneous fungi, right? At a word pohibo lagibo ami, but if someone has read, you have come across this word that is called as heterogeneous. Heterogeneous fungi mane ki je jikono eta fungi jedi duta hostot life cycle to complete kore, tene tak ami ki boli ko heterogeneous fungi. Generally, whenever we are talking about a pathogen, hey pathogen to ki koribo life cycle to eta hostote complete koribo. Means what? This pathogen will affect one plant and that is the entire life cycle, no? Hetu ahibo plant dalot reproduce koribo, increase in number koribo, tapsot spread koribo. So its entire lifespan is revolved around one host. Kintu kisuman enuka type of pathogens thake jar niki tumar host duta thake, right? So jedi duta hostot tar life cycle to complete hoy tetihal tak ami ki boli kom heterogeneous. Details about heterogeneous fungi again in the portion of fungi when we will discuss. As of now, we'll come back to this. Black rust of wheat kune cause kore, Puccinia cause kore. One point of Puccinia that you need to know that it is an example of a heterogeneous fungi. What is a heterogeneous fungi? A fungi that completes its life cycle in two hosts. That is what is called as a heterogeneous fungi. Next is bacterial blight of rice. Ako causative organism important, Xanthomonas oryzae. So bacterial blight of rice, bacterial word to already present. So this time what we know that this is a disease not caused by fungi, but instead it is an example of a bacterial disease. So causative organism ki hobo? Causative organism eta bacteria hobo, right? Similarly, the next is cucumber mosaic disease. Cucumber mosaic disease. Cucumber we understand. Mosaic ki hoy? What is mosaic? Mosaic in simple words what we understand is patch, no? Eta bostud by eta patot jedi certain types of patches that you see, that is what is called as mosaic, right? Different colors in the same leaf, different colors in the same stem, right? That is what is called as mosaic. Uneven structures by uneven colors, right? So cucumber mosaic disease kune cause kore? We have to remember it is caused by a virus. And what is the name of the virus? It is cucumovirus, right? Virus of kotha porile, again we have to talk either it is RNA virus, ne DNA virus, single stranded virus, ne double stranded virus. So in this case we have a single stranded RNA virus and the name of the virus is cucumovirus. Next is the last one is root knot of tomato. Etur pora birat questions ahise. Actually gutei gita important. Ihar pora ahise, etur pora ahise. Root knot of tomato and brinjal is caused by a nematode. It's not bacterial, it's not fungal, neither it is viral, no? This is a disease that is caused by a nematode. You have to remember the name of the causative organism, Melogdigni incognita, right? So plant breeding for disease resistance, juar agote, we have to talk about certain types of disease. Egita disease you need to by heart. Poribo lagibo, causative organism kita janibo lagibo, what are the symptoms? In it just for GK sake you need to know. Now, in order to make a plant disease resistant, ami ki koribo lagibo, we have to go for plant breeding, right? So disease hole to crop loss hobo, crop loss no hobo lagile, amak ki lage adal plant lage, arjundal plant disease resistant hobo lage. Jedi disease resistant plant adal ami produce koribo lage, then what we'll do? We will go for certain methods like conventional breeding and mutation breeding. Conventional breeding is something that we have discussed. If you remember, steps of plant breeding, I mean discuss korisu no? Kiki steps asile. We have talked about that collection of variability, then we have talked about selection of parents, right? Testing for superior recombinants, and then ultimately it is testing, releasing and commercialization, right? So those are the steps that falls under conventional breeding. And what we know, conventional breeding is a very lengthy process, right? Time consuming process. So in order to create a disease resistant plant, either we can go for this conventional breeding or we can go for the mutation breeding. I think you remember we have already said that if you are talking about a faster type of a program, then obviously, or the most advanced type of breeding is obviously nowadays it's mutation breeding, right? Mutation breeding koribo lagile iman besi steps ami follow koribo nalage, right? Kisuman physical ba kisuman chemical agents apply kori pelai, we can go for inducing mutation in those plants, right? So ultimately we go for mutation breeding. Another point that we do not go for conventional breeding. Ami conventional breeding to generally etia kiho karone apply nokoro. The first point is it is a time consuming process, right? Time consuming process. Second is yar karone first step ki je ami germplasm to bisaribo lagibo. Aru in case of this germplasm bisarato problem, disease thaka plantor germplasm bisaribo lagile, this is a problem, and I think there is a disturbance, right? What? Someone is causing a noise. Okay, can you hear me now? Hello? Okay, we'll come back to this. So what we know is conventional breeding is not used. What we are using is generally we use nowadays mutation breeding. So we had an idea from this part. Next is, as I have said at the starting of this lesson itself, what I said is the most important portion of this lesson is table 9.1 and table 9.2 of your NCERT book, right? So NCERT book dukon table ase, hey dukon table of maximum questions from this chapter has been asked. Either it is board or it is in your MCQs. So we'll be talking about table 9.1 and 9.2. 9.1 dot ki ase or 9.2 ki ase. In this table we have resistance to disease, and in this table we have resistance against insects, right? Tamane ki we have made certain types of resistant plant, right? Plant breeding koriso, plant breeding kori pelai what we have prepared is certain types of resistant plant. Now this plant is resistant against what? It might be resistant against diseases or it might be resistant against certain types of pests. So accordingly, resistance against disease or resistance against pest. Come to the first one. Tate ki diya ase? Sarita different type of plant diya ase. We have the crop, we have the variety, and we have the resistance to disease. So this chart is very important, right? Birate important because maximum questions here pra ahe de. So yate question keneke ahibo. For example, if you are talking about wheat or a variety, the variety is called as himgiri. Himgiri is resistant to two types of disease, leaf and stipe rust or hill blunt. Now A2 question ahana is what is hill blunt? So you don't have to worry about that you have to read about those type of disease, keneke hoy symptoms ki, you don't need to read about that, right? Just remember the names. Question keneke ahise? Question ahise, the himgiri is a variety of, right? So himgiri is a variety of, in the options you might have all this, and so in that case what you need to choose is himgiri is a variety of wheat. But question to vice versa ahibo pare, so wheat or variety kuntu, wheat or variety pusa swarnim hoy, pusa shubra hoy, ne himgiri hoy. So this is a column that you need to remember, right? Second is, question from this part is also been asked. What is asked is, hill blunt is a disease that is resistant to dash variety of wheat, that is resistant to himgiri variety of wheat, or himgiri variety of wheat is resistant to dash disease. So you need to remember the disease also, right? So you don't have any other alternative instead of by hearting these two charts, right? A2 chapter or A2 chart to be mukhosto koriboi lagibo. Questions I say. Similarly, if you are talking about brassica, I think we remember what is brassica. We are talking about the family called as Brassicaceae, and whenever we talk about Brassicaceae, the best example that we remember is Brassicaceae at an example kwa. Just give me an, yeah, it could be cabbage, it could be mustard, it could be cauliflower, right? So these are examples of Brassicaceae. We are talking about Brassica. In this Brassica, ako pusa swarnim and white rust, right? So ekini bostu ami porim. Cauliflower you are talking about, you have to remember shubra and snowball. If you see, pusa is something that is common to all the, from 2nd to 5th, pusa is something that is common. What you need to remember is swarnim, shubra, komal, aru Sadabahar, right? Generally, in this chart, question maximum ko pra aise, wheat or question aise, or chili question aise. So, eduta part ami bhalke porim. Next is, what is a causative organism? Again, CMV, TMV, leaf curve. TMV, I think we have an idea. What is TMV? TMV ki hoy? Short form for? It is a viral disease. Ki asile? Full form to? Tobacco mosaic virus. Right? So, this is tobacco mosaic virus, this is chili mosaic virus and leaf curl. এই তিনটা ডিজিজৰ পৰা resistant হৈ আছে কোন resistant হৈ আছে a variety of chili and what is the name of the variety pusa sadabahar তাৰ মানে এডাল প্ল্যান্টত এই তিনটা ডিজিজ কেতিয়াও নহয় is the point understood যেনেকে হিমগিৰিত এই দুটা ডিজিজ নহয় ঠিক তেনেকে ইয়াতো এই দুটা ডিজিজ নহয় similarly pusa কোমলতো bacterial blight নহয় so এখন chart আমি মুখস্থ কৰিম next coming to the second chart that is table 9.2 table 9.2 কি দিয়া আছে resistance against কি দিয়া আছে insects yate ki diya asile resistance against disease means what there are certain insects which cannot invade or which cannot attack certain types of plants right so if you are talking about an insect called as aphid kak attack koribo nware pusa gaurav variety of brassica beleg variety of brassica kintu attack koribo right okol etu variety aphids pula insect to tak attack koribo nware jedi attack koribo nware obviously that disease will not be caused by that insect right similarly if you are talking about flat bean you have certain insects called as zacids aphids and fruit grower that cannot infect these two varieties of flat bean similarly shoot and fruit borer cannot infect okra right Ekon chart opera key question is jeneke mo kolu from this chart what you need to remember is the variety and the disease is something that the question is asked about from this chart what you need to remember is the name of the insects ekhini birat important right question to ki ahe the brassica is resistant to which disease which insect teneke pusa gaurav is resistant to which insect but aphid is resistant to which variety right thik teneke zacids is resistant to flat bean generally from this variety part ekhon chart opor question aha nai question ahise about this part crop portion to aru insect portion to kintu ekhon chart ot tumi gutei tinta bostu monot rakhibo lagibo right so up till here what we have concluded is we have been talking about disease resistance or plant breeding for disease resistance so ultimately what we have produced is we have produced certain type of plants that are resistant to certain types of disease and certain type of plants that are resistant to certain type of insects right so this is all about this topic kiba doubts asile okay So after this, the next topic that we are going to discuss is something called as biofortification. Have you heard the word called as biofortification? There are certain words like biofortification, biomagnification, right? So yate ami ki discuss kori biofortification. What is biofortification? Ta mane ki? Ami ki pori aso? We are talking about plant. Yeah, so we are talking about plant breeding, right? So, erukon chart ot ami ki poriso breeding for diseases, disease resistance, breeding for insect resistance, etc. Now we would be talking about disease for improved food quality of food, right? Atake ami ki boli kom biofortification boli kom. Ki hoise? Side kon. So after reading this part of today's chapter, what you do is just try to find out what are the type of questions that have been asked from these two charts, right? So go through your MCQ books and see whether you could answer or not, right? So you need to do certain types of homework also. Okol pori gole etu nohobo, hoy na hoy? Okoman MCQs book koriba until and unless you open the MCQ books, you will not have an idea that what is the type of question that is asked. Right? Okay. Okay. The next topic is biofortification. fortification, right? So biofortification, what we know that what we are doing is that there are certain foods, no? Hey, food be like, I mean actually food kiyohor karone khao, so that we can get the essential nutrients. And what are the nutrients we are talking about? We are talking about proteins, vitamins, minerals, micronutrients, etc. right? Kintu ami jobilak food khao, not necessary that they are rich in all the essential nutrients, right? So we have seen that many people or billions of people are suffering from something called as hidden hunger, right? So you need to remember hidden hunger. So what is hidden hunger? Hidden hunger is a term that is used when suppose a person is having a diet, according to him he is having a proper diet, kintu hey proper diet on jodi essential nutrients bilak nathake tetia hole that at that time we use the word hidden hunger. So billions of people are suffering from hidden hunger. Why? Since their diet is poor in essential nutrients like proteins, vitamins, minerals, micronutrients and fats. So no doubt they are having the food, but the food is I mean it's lesser in quality, and so at that time we have something that is called as hidden hunger. Etia enukwa dhoroner diet khai pelai to lav nai, no, you should have a diet that is proper in your nutrients. Aru if we do by some process ami jodi food or quality to increase koribo paro, that process is called as what? Biofortification. So what is biofortification? It is the enhancements or it is the increase of the quality of proteins, minerals, fats, etc. in the diet, right? So biofortification mane ki increase the quality of proteins, vitamins, minerals, fats, etc. in our diet, right? So jodi ami increase koribo paru tetia tak ami hey process to ami ki boli koiso biofortification. Biofortification is a process through which what will happen that the quality of the food will increase, right? Aru ki increase koraiso, so we are talking about all these nutrients, right? So what is the objective of biofortification? The objective of biofortification is to increase or to enhance the quality of food. From this part, ako kisman points ami monot rakhibo lagibo. From examination point of view, it will be question ahe. First question ahe what is biofortification? Definition wise, that is something that we need to remember. Next is what is the objective of biofortification? To enhance the food quality. Next is kita point. Cereals are deficient in lysine and tryptophan. Right. Aru pulses are deficient in sulfur rich methionine and cysteine. Right? So biofortification jehetu kotha koi aso, the food, one of the major foods that we take is obviously cereals and pulses, no? That is supposed to be a staple food. So what we have seen is, or what did the scientists see is, when we are talking about cereals, what would be the example of a cereal? Cereal or example ki? Pulse buli kole what we understand, cereal buli kole what we understand. Cereal example rice, wheat, etc. These are examples of certain types of cereals, right? If you are talking about pulses, the pulses could be the different types of dal that we have, right? Lentil, that is an example of a pulse, right? So cereals belong normally kisuman amino acids or deficient hoi thake. And what are the amino acids we are talking about? Lysine and tryptophan. Lysine, tryptophan, methionine, cysteine. All these are nothing but certain types of amino acids. And why is amino acid essential? Food or basic amino acid thakile ki labh hobo? যে আমি যেটো ফুড খাইছো তাত অকমান অ্যামিনো অ্যাসিড বেসিকে থাকিলে it's better for us. Why? Because we know that amino acids ultimately will form what? Proteins, and proteins are obviously the basis of the cell, right? Backbone of a cell বা most important constituent of a cell কি হয়? Protein এটো হয়, right? So proteins বিলাক কোনে ফর্ম কৰিব? Amino acids বিলাকে ফর্ম কৰিব. So when we are having these foods that are deficient in amino acids, that type of a food is considered to be a low quality food. So ইয়াতে question কি আহে যে directly এই দুটা লাইনৰ পৰা আহে যে cereals are deficient in, or question to ulta ke ahibo pare, lysine and tryptophan deficiency is seen in, it's seen in cereals, pulses, millets, etc., etc. So ultimately the answer would be cereals. Similarly, if you are talking about pulses or the different type of dal that we have, those are deficient in sulfur rich methionine and cysteine. Methionine or cysteine ki hoy? These are again some examples of amino acid. So yate kisuman amino acid, yate kisuman amino acid, and these 2 points are important from the examination point of view, right? So eto lysine or upor birat question ahe, and methionine or upor to both questions ahe, right? Next is because with the motive that we need to increase the quality of food, no? Etia kisuman plants bonua hoise ba plant breeding or through there are certain plants that has been made which are high in food quality. So when we are talking about such type of plants, we need to give some examples. Proteina, Shakti and Ratan, right? Next is tomato, mustard. Then we can talk about high iron, spinach, right? And these are certain plants that are developed by IARI. What is the full form? Indian Agricultural Research Institute. Our port of it is in New Delhi, right? So what we are talking about is biofortification. Biofortification at ami ki koru je food or quality to ami increase koru. By increasing, what we understand is we increase the contents of the most important product of the food, that is essential nutrients like proteins, minerals, vitamins, ebilak bostu ami increase koru, right? Next is, so since we are talking about plant breeding, there are certain types of plants that has been newly created which has high source of some essential nutrients. For example, if you are talking about maize variety, you know, kisuman maize variety eneke produce kora hoise jot niki tahator ki thake lysine content to besi thake. Olop agote what we have discussed is whenever we are talking about cereals, then we have seen that cereals are always deficient in 2 types of amino acid, that is lysine and tryptophan. So what did the scientists scientists do, they have created a plant that has high rich of lysine. So examples of maize that has high rich of lysine is Protina, Shakti and Ratan. Ako names are important. Similarly, there is a high protein wheat also, normal wheat or a high protein wheat, right? Wheat or high protein variety to naam ki, it is Atlas 66. Ako etu namu monot rakhibo lagibo. Similarly, scientists from time to time, they have been trying to improve the quality of food. And who is conducting all these experiments? It is done in something called as IARI, or Indian Agricultural Research Institute, that is in New Delhi, right? So that ki hoy se normal carrot to ase tar upor high vitamin A thaka carrot to bonaise thik teneke normal pumpkin ase kintu high vitamin A thaka pumpkin. Similarly, we know spinach is a rich source of iron kintu tate iron content to borhai pelai, the crop has been modified, right? So similarly, if you are talking about vitamin C, increased amount of vitamin C is seen in some varieties of tomato and mustard, and who is doing this? They are really responsible for increasing the quality. Right. So this is something about biofortification that you need to remember. Here we got important key definition and the examples of the varieties. Right. Basic tan part no hoy kintu okoman mukhosto koribo lagibo. So what else is present in this chapter? Biofortification we have done, plant breeding we have done. The next is single cell protein. Single cell protein, it is also called as SCP. It is denoted by SCP. So what is a single cell protein? Single cell protein boli kole, what we understand, there are certain microorganisms that we use them as food, no? And these are considered as single cell protein. Filamentous algae, etc. are used as food source. Certain microorganisms like bacteria, fungi, filamentous algae, etc. are used as food source, and these are called as single cell protein, right? So what is single cell protein? The kisuman unicellular organisms ase jun bilak niki ami food hisabe use koru aru tahatok ami ki buli ko single cell protein buli ko. Single cell protein koribo lagile some points that you need to remember. Raw materials kisuman lagibo, and what are the raw materials? Whey, molasses, straw, wastewater containing starch, etc. Right? So single cell protein, কোনবিলাক single cell protein যোনবিলাক নেকি এনেকুৱা ধৰণৰ microorganisms যোনবিলাক আমি খোৱা বস্তু হিচাপে ইউজ কৰো, those are called as single cell protein. So we need to produce them also. So generally process to keneke hoy that we go for fermentation, fermentation process pora with the help of fermentation these are allowed to grow and multiply, and ultimately we use them as food. In order to produce in huge quantity, obviously raw materials kisuman lagibo, and so these are supposed to be certain types of raw materials that are used for this process. Next is, what is the process that we used? Generally we use by the process of fermentation, or they are produced in they are produced in large fermenters or fermentation tanks where they are aerated for vigorous growth, right? So as I said, single cell protein or karne kuntu process important, it is done by fermentation. Fermentation tank kisuman thake aru he tank bilakot ki hoy tahatok ami grow koribol diu ba multiply koribol diu, right? So for this purpose what we do is we put those single cell protein in that fermenters. Our fermenters, what we do is we apply a process called as aeration. Aerated for vigorous growth. Aerated by aeration, what we understand, যেটা ফার্মেন্টার থাকিব, ফার্মেন্টার বা ফার্মেন্টিং ট্যাঙ্ক এটা থাকিব, ট্যাঙ্কত কি থাকিব কিছুমান ধৰণৰ কেমিকেলছ থাকিব, প্লাস ইউ হ্যাভ দোজ সিঙ্গল সেল প্রোটিন, আৰু তাত তেনেকুৱা ধৰণৰ ফার্মেন্টাৰ্স বিলাক, সাপোজ দিস ইজ আ ফার্মেন্টার, ইয়াতে ইউ হ্যাভ দ্যাট কেমিকেলছ, ইয়াতে সিঙ্গল সেল প্রোটিন, ইয়াতে কি থাকিব যে কিছুমান এয়ার পাম্পিং এটা এরিয়া থাকিব, রাইট? সো ইয়াতে আমি কি দিও, we apply air from outside and that is what is called as aeration. So if suppose you all remember something called as itu portion etilage porpa nai, tothapi we'll be talking about sewage disposal. Sewage or upor ami jetia porim, we'll be talking about fermentation again. As of now what we understand is single cell protein fermentation kore aru fermented tank bilakok air is supplied from the outside and as a result jetia oxygen pabo tetia hole ki hobo, aerobic organisms bilakor they will grow and multiply, and this is what we wanted, no? Jehetu ami khwa bostu hisabe use korisu, we always want that their number should increase, right? So in order to increase, one of the method that we do is we go for aeration method in the fermentation tanks, right? Next is, what are the examples of plants that are used as single cell protein? So yate ami examples kisuman monot rakhibo lagibo. If you are talking about algae kisuman algae we take them as food. Spirulina, chlorella, scandesmus, etc. So these are the algae that are used as food. If you are talking about fungi, Candida. Next we can talk about yeast, etc. So if you are talking about bacteria also, we are talking about Methylophilus bacteria, etc. So microorganisms used as food is called as single cell protein. Certain types of algae that is used as food, certain type of fungi that is used as food, and certain type of bacteria that is used as food. Out of this, the question has maximum been asked about the examples of algae and obviously about yeast. So baki kita examples of algae or examples of yeast is something that you need to remember. Now single cell protein bilak ami food hisabe use korisu, no? Use koribo lagile there should be certain types of advantages also. So what are the advantage, right? Normal food khwar single cell protein for advantage ke, the advantage is of a single cell protein is, first is higher rate of multiplication, right? So a single cell, eta cellok jodi ami food hisabe khao tahator ki hoy, multiplication rate is always faster, no? I mean we have been talking about the reproduction method in case of unicellular organisms. So higher rate of multiplication, as a result higher rate of food production, right? That is the first one. Second is tahate they are not dependent on any type of climatic variation. Jeneke amar plants bilak, normal plants bilak jibilak ami food hisabe khao, they are always dependent on climatic variations. For example, they will have a particular season in which they flower, right? Flower to ripen hobo lage, ripening season eta thakibo, harvesting season eta thakibo. So they are always dependent on environmental variations ba climatic variations. Kintu single cell protein as a food jodi ami use koru, teta hal tahatok kono dhoroner environmental mane variations ba climatic variations or karone karne we don't have to depend upon, right? So in any type of climate that we produce, they are capable of multiplying in a very faster rate. And the third advantage could be, it is like, first আমি কি করলো, যে high rate of multiplication, second is resistance or independent of climatic variation, third is genetic modification is easier, right? Eta single cell protein ase, he eta particular type of food prepare korise, and what we want is, we want a different or a good quality of food that it produces. So in that case, hey single cell protein to genes bilak ami jodi modify koru for our use, tete hale ki hobo, tar production to bari jabo. Genetic modification or through re, we have been talking about mutation breeding, right? So genetic modification or through re ami ki koru, better plants for food ba better plants for disease resistance ami produce koru. Similarly single cell protein bilak kok jodi ami better koribo lage, tete hale ami tak ki korim, genetic modification or karne jam. And whenever we are talking about genetic modification, then obviously genetic modification in a unicellular organism is always easier as compared to that is done in case of a multicellular organism. Karon unicellular organism or ki thake, etai cell thake, eta cellot jiman kini nuclear material ase, that would be easy. But in case of multicellular organism, iman bur cell ase, protective cellot nucleus ase, protective nucleus of the genetic material ase, so that is a hazardous work, right? So these are the three factors, or these are the three advantages of single cell protein, right? So ki asle advantage, the first is high rate of multiplication, as a result ki hobo, high rate of multiplication hole high food production of food hobo, no? Second is environment ba climate opor depend nokore, right? Rainy season, winter season, summer season ebilak eku kotha nai. Third is genetic modification easy hobo. So these are the things. Yeah. Yeah, the fourth is, you can talk about, it reduces environment pollution also. Okay, what we understand, that's what we understand by it reduces environmental pollution at a simple way is, if suppose we are talking about an animal as a food, right? If we are talking about this, for example, we are talking about chicken, right? So chicken eta tumi ki koribo lagibo, tumi tak bhalke rakhibo lagibo, khuabo lagibo, this that you have to do, right? You have to make it grow properly. Plus tar logote ki hobo, since it's a living being, it will tar excretion hobo, waste products to produce koribo, right? Hey, waste product of that chicken kot jabo, environmental le jabo. So as a result, environment pollution holle nai. That is in case of animal food. For example, in case of plant food also, adult plant as a plant or to be food to lola, but you take the part that is essential. What happens to the rest of the plant? The rest of the plant gets decayed. When it gets decayed, in one sense, it causes environmental pollution also, right? Eta jega lagibo joti ki tumi hekini putiba ba hekini geli jabo. So these are the sources of environmental pollution. But since this is a single cell protein, environmental pollution kora tar kono chance nai, karon tar waste product e nai, no? Waste product ase niki. We have talked about that unicellular organisms are generally considered to be immortal, no? I think we remember this sentence, no? So jehetu they do not die, so almost they do not die, no? And what is the meaning of this sentence? We understand. Jehetu tahator hetu no hoy, so they live as their daughter individuals. So the waste product produced korato, it's almost nil. So that is the reason why we say they reduces environmental pollution, right? Okay, welcome. So single cell protein. Now pisot, the next topic is green manure. Right? So what is green manure? When you grow back the young plants back into the soil, it is called as green manure. Tamane ki kisuman plants ase, hey plants bilak ami ki koru, ughali pelai akko mati puti diu, right? In that case, the sentence that we are using is, we grow back the young plants again into the soil. Grow back the young plants into the soil, right? So young plants bilak akko jodi matit ami use koru ar tak ami ki boli kau, we are talking about to prepare green manure, right? Next is, for this purpose, what are the type of plants that are used? Generally kunbilak plants use kore for green manure, it is always leguminous plants, right? So green manure hisabe kunbilak plants use kore leguminous plants bilak generally green manure hisabe kyo use kore why not the other plants leguminous plants has an advantage and what is the advantage we are talking about it is rich in what it is rich in nitrogenous substances right so leguminous plants rich in nitrogenous compounds no? Or kiho karone they are rich in nitrogenous compounds yeah how they are rich in nitrogenous compounds if you remember leguminous plants or logot we always talk about nodules no root nodules or kotha patun no so these root nodules or nodules are the site of key these are the site of nitrogen fixation so if they have enough nitrogen and that comes back to the soil obviously that area will become more fertile right so leguminous plants they are rich in nitrogenous compounds because they have nodules and nodules bilak ki hoy fixed atmospheric nitrogen right so kisuman plants ami manure hisabe use koru tar mane ki we make those plants increase the fertility of the soil that is what is something that we understand by the word manure kun bilak plants use koru leguminous plants bilak use koru kiho karone use koru because they are rich in nitrogenous compound now there are certain examples of plants that are used as green manure and these examples are examples of green manure or examples of plants that are used as green manure Crotalaria that is sunham Sesbania sweet clover cowpea lentil etc right so there are certain plants that are used as green manure for example sunham sesbania sweet clover cowpea etc out of this maximum question has been asked from this point and this point right sesbania or crotalaria in case of sesbania only this name is sufficient to remember but in case of the first example common name or scientific name crotalaria jansea right but crotalaria okol generic name to monot rakhile hobo so question has been asked about the common name also and about the scientific name baki rest of the things jikhini disu that's more than enough tatke upor aru tumar scientific name na lage so sunham crotalaria and these are the examples tar pitor important kuntu obviously crotalaria is something that is important right eplagor pro ako ami examples kini monot rakhim now what is the advantage of green manure but green manure ami use korile Kisman advantage ase no what is the advantage or the greatest advantage is it increases the fertility of the soil increases fertility of the soil keneke increase kore karon nitrogen ase besike mane hol yeah and nitrogen is something that is very much essential for the plants second it also reduces alkalinity and it also prevents soil erosion alkalinity alkaline hoi thakile plants bilak bhalke grow nokore right plants bilakok so eta proper eta pH balance thakibo lagibo no right so যদি alkalinity by eta soil etukura soil ase which is alkaline in nature tetiale he plants bilak grow koribole problem hoy in that case that feature needs to get reduced aru okoman acidic type ole convert koribo lagibo so kune help koribo ebilak plants e help koribo right so these are the advantage of green manure and as a result ki hoy by application of green manure we have seen that junbilak area ami green manure use koriso tat ki hoise crop production has always been increased by 20 to 30% right So instead of using the artificial chemicals as manure, right, it is always advisable to use the green manures as something for increasing the fertility of the soil. So this is all about green manure. So aji ami ki korilu, biofortification is something that we have discussed, right? Disease resistant, insect resistant. Next, single cell protein, right? And the next topic that we are going to discuss is energy crops. So what we understand by the word energy crops, right? So scientists has been trying hard to produce alcohol from plants. So scientists they are conducting various experiments so that they can produce alcohol for plants. And why do they need this alcohol? To run the automobiles, right? Petrol, diesel, ever use nokora ke ami jodi kisuman tenukwa dhoroner product kisuman plants or pra use koribo paru ba ulabo paru. This is what is called as energy crops. You get those type of energy or you get those type of products from certain types of plants. Our alcohol, what they are trying is to run the automobiles. Setya hole ki hobo, petrolor demand to naikia hoi jabo, because petrol, as we know, first it is very costly, second tar now amount is decreasing gradually, right? So setya ekebare naikia hoi jabo, tetya hole gari bur keneke cholibo. So we always need an alternative, and what is the alternative that the scientists are working upon is to produce such type of compounds from certain type of plants, and those plants are called as energy crops, right? So various programs has been conducted, and when we talk about these different programs that has been conducted in the world, we have to talk about some of them. For example, there are certain programs that have been conducted in Brazil and some in USA also. Right. So when we are talking about Brazil ki korise, they are conducting a program called as Pro Alcohol Program, right? And what they are doing to with this program, what they are trying to do is replace petrol or gasoline with alcohol. Right? So tahate enuka dhoroner pro alcohol program eta kori ase. Plus they are also trying to extract alcohol from sugarcane. So these are the various type of experiments that they are conducting to take out alcohol from certain types of plants. Similarly, if you are talking about USA, they are also conducting certain types of experiments or programs. They have a program called as gasohol program, and they are experimenting with the plant maize or pora alcohol ulabole tahate experiment kori ase. And as far as this program is concerned, within this program they are trying to blend 10 to 15% of alcohol with petrol. Right? So energy crops, that is a very new technique. So there are certain countries that are doing experiments on this. So these are the programs that we need to talk about, right? Certain types of plants or alcohol extract koribole chesta kori ase. Next is, what are the plants that are being used for the production of alcohol or are being experimented for the production of alcohol? Right? So kunbilak plants of kami generally energy crops bilak ko junbilakor pora alcohol bilak ulabo. For example, kunbilak plants, we have sugarcane, maize, potato, sugar beet, etc. So eblak plants or pra ki korise alcohol ulabole chesta kori ase. So we have a term called as energy cropping. So what is energy cropping? Production of alcohol or trying to produce alcohol from certain types of plants is called as energy cropping. And what is the reason behind? So that we can depend on these plants for running of the automobiles. Now this type of a program has certain disadvantage also. What are the disadvantage? Enuka dhoroner bostur kisuman to advantage thakibo. Advantage ki je petrol ami replace koribo parim alcohol. They have been trying to do that. And there are certain types of disadvantage also. Iman popular kiho karone hua nai. Right, because scientists has already extracted alcohol from certain types of plants, but in spite of that, this program has not been very much commercialized. The reason behind this, the certain types of drawbacks. And what are the drawbacks we are talking about? First is very much cost. It's very costly. Enuka dhoroner program eta koribo lagile plantor pora alcohol ulabo lagile, it is very costly. Our cost factor is one of the disadvantages. Second is loss of agricultural land. Loss of agricultural land mane ki junkhini jagat ami proper crops grow kori asilu, that crops that were meant for food, no, instead of that crop what we are planting is those crops from which we can take out the alcohol. So as a result agricultural land bilakor loss hoise. Loss in the sense যে য'ত suppose for example য'ত rice হ'ব লাগিছিলে তাত আমি rice নুৰুই পেলাই কি ৰুই দিলো potato ৰুই দিছো আৰু potato পৰা আমি কি কৰিছো we are trying to take out the alcohol. So these two are the major disadvantages of energy crops. Now whenever we are talking about energy crops, crop gita monot rakhiba ni and the term also, we have to talk about something called as petroleum plants. But petrol plants. Right? So yate ami ki jano je a scientist called Melvin and Kevin, sorry. Right? From time to time what they have suggested is that they can extract petrol like substances from certain types of plants, right? Kisuman plants or pra ki koribo pare petrol or nisina bostu kopra ulabo paribo, kisuman plants or pra ulabo paribo. And which plants they said that these plants should produce or should have large amount of latex. So kisuman enuka dhoroner plants ase junbilak plants e huge amount of latex produce kore. Now what is latex? That gum or sticky like substance is what we are talking about, right? So they are of the opinion that there are certain plants, those plants that produce large amount of latex, tahator pora ami ki koribo paru, we can take out the petrol, right? Or these are called as petroleum plants. Now why latex? Because according to them latex or ki thake eta tumar it's a long chained hydrocarbon by long chain liquid hydrocarbon latex thake ar petrolot ki thake petroloto eblak enuka dhoroner chained hydrocarbons thake. So what they have noticed is a similarity between petrol and the product that has been extracted from this plant, and so they are of idea that eblak plants or pora ami petrol type of a substance or petrol ami extract koribo parim. And since we are trying to take out the petrol from the plants, these plants are called as what? Petroleum plants. Now what is the question that is asked from this part? Petrol plants by energy crops or jimandu examples discuss korilo, maximum the question has been come about this petrol plants. So enuka kunbilak plants. So what is the plants that they are talking about? These plants generally belong to families, right? So petroleum plants bilak kunbilak families are generally dekha pai, they belong to the families like Euphorbiaceae. Second could be Asteraceae. Third could be Apocynaceae, etc. Right? So junbilak plants e basically latex produce kore, hebilak plants or pra petrol type of thing extract koribo pare. Aru ebilak plants kunbilak families or poa jai. So these families are those that are responsible for something that is called as petroleum plants. So what are the families? Euphorbiaceae, Asteraceae, Apocynaceae, etc. Out of this example, question has been asked about Euphorbiaceae, right? Euphorbiaceae is a family. One example of Euphorbiaceae could be Euphorbia, right? So aru ekhini hol families aru plants or jodi example dio tetia hole one example could be Euphorbia, the second example could be Xatropha, right? and Jatropha report birat question ahise. Question to ki ahise, identify the petroleum plant or the plant that is responsible for the production of petroleum, or ketiaba ki hoy energy crop or within to heading kori pelai etu question ahe, identify the energy crop. So out of all the examples that we have remembered from entrance point of view or from examination point of view, Jatropha is the most important example that we remember, right? So up till here, so this is the things that we have discussed today. Right. I think this is enough for today. What we have discussed is, we are just summing it up. I mean, first ki discuss korilu, disease resistant, right? We have talked about plant breeding. So kisuman plants ami disease resistant hisabe banao, aru kisuman plants are there which are resistant to certain types of insects. What is important? Table 9.1 and table 9.2, right? You have to by heart. After that, we have talked about biofortification. Biofortification, what is important? Definition of biofortification, right? Second, the most important point is cereals are deficient in and pulses are deficient in, and some examples of biofortified plants, right? Next is single cell protein. Again, examples of single cell protein, right? What are single cell protein? Those single cell microorganisms that we use it as food, right? Next is something called as green manure. Examples of green manure and which are the plants? We have to remember the word leguminous plants. Right? And lastly, we have discussed something called as energy crops. Whenever we talk about energy crops, maximum stress has been given on the two words Euphorbiaceae or Euphorbia and Jatropha. Right? Do you have any doubts from this section? Okay, so please go through the lesson properly, and in the next class we will be discussing the last part of the chapter, that is tissue culture. Right? Thank you for today.